So I will pause there and you can note down, because this is all very very important for your other competitive exams also. So whatever you want to note, you can note down. Cellular respiration is a complicated process, children. It takes time in understanding. और एक बार अभी पूरा आपने पढ़ लिया, आप थोड़ा सा और डिटेल में जाएंगे तो you will be able to understand further. ठीक है, स्टार्टिंग में अगर हम उतना डिटेल बताते तो आप नहीं समझ पाते. Okay, but now since you have understood the whole thing and you have prepared also, so things are more clear. So now try to understand. My screen is visible to you all.
Yes, ma'am.
Okay, so now I'm playing the tricarboxylic acid cycle. What is it? Glycolysis, तो I hope everyone has understood. अगर glycolysis भी एक बार और revise करना है तो you tell me. So I will show you that also once again. Okay, but we will start with the Tca cycle and the electron transport system.
Citric acid cycle. The first stage of cellular respiration is glycolysis. In this process, glucose is broken down to form a 3-carbon compound, pyruvate. In the presence of oxygen, pyruvate enters the mitochondria where it undergoes decarboxylation to form a 2-carbon compound called acetyl coenzyme A. Acetyl coenzyme A enters a set of cyclic reactions by combining with the 4-carbon compound oxaloacetate to form a 6-carbon compound, citric acid. As citric acid is the first product of the cycle, this cycle is called.
Children, the volume is okay for you all?
Yes, ma'am.
Okay, all can hear well now? ठीक है, तो first step समझ में आ गया, that is your oxidative decarboxylation, coenzyme A बन गया, further entering into the Krebs cycle. How it will enter? To ester coenzyme A combining with the oxaloacetic acid or oxaloacetate. So it's showing with the figures very clearly. ये 2 carbon वाला है, ये 4 carbon वाला है. And when these two will be combining, so then you will find the first compound form will be the citric acid. And this citric acid is having 3 carboxylic group, इसलिए अपन उसको tricarboxylic acid cycle बोलते हैं. That is Tca cycle, and it is also called citric acid cycle because first compound form is citric acid, and other name the Krebs cycle because hence Krebs has studied all these reactions. Okay, that is the cyclic reactions. क्योंकि सारे reactions होने के बाद you find at the end we get the oxaloacetic acid again. That is oxaloacetic acid is regenerating. So it is the reaction is going on in a cyclic way. So you call it a cycle. Tca cycle or tricarboxylic acid cycle or citric acid cycle or Krebs cycle. Is it very clear to everyone this much? Now see the reactions nicely.
Citric acid cycle. It is also referred to as the Krebs cycle, named after its discoverer, Hans Krebs. In this cycle, citric acid undergoes a series of cyclic reactions to regenerate oxaloacetate. The energy released during the cycle is transferred to cofactors such as NAD and FAD. In one of the steps, ATP is formed directly. The reduced cofactors NADH, H+, and FADH2 enter the electron transport chain. The energy stored in these molecules is converted to ATP. Oxygen is the final electron acceptor. It combines with protons to form water.
Is it clear now to everyone this much?
The energy stored in the pyruvate molecule is extracted in the 5 stages of the Krebs cycle. Click a tab to learn more.
So one by one, each step you see.
On entering the mitochondria, pyruvate undergoes oxidative decarboxylation to release a molecule of carbon dioxide and form a 2-carbon compound called acetyl CoA. Also, NAD+ is reduced. The pyruvate dehydrogenase enzyme complex catalyzes this reaction.
So children, those who want to write down this equation, you can note down. Okay, this is pyruvate, which is a 3-carbon compound. So you can see here COO, then CO, CH3. So to this pyruvate, the coenzyme is combining. So you can see here the coenzyme A combining, and you get acetyl coenzyme A, and carbon dioxide is removed. So the 3-carbon compound is becoming 2-carbon compound now. Anyone noting this? Yes or no? If anyone noting to say, otherwise I will take up the next slide. Neha, Barbie, speak up children, unmute your mic.
Ma'am, already noted.
Are you all noting?
Yes, ma'am.
हाँ, anyone, हाँ, those who want to note, क्योंकि ये सब आपको क्लियर होना चाहिए other competitive exams के लिए. Okay, so if you wish, you can write down. जिसको लिखना है वो जल्दी जल्दी से copy कर लो. One reaction it will be showing. Okay, because that time when we were discussing this cycle, I have not told you their formulas. Okay, so now you can note down all that. As you complete, let me know. This is which process going on? This is the reaction for children. What is it?
Oxidative decarboxylation.
Correct. Oxidative decarboxylation. Okay, shall I take up the next now? I'm changing now. Done.
Yes, ma'am.
Yeah, so you have to say that.
Click a tab and click the correct slot.
Okay, this we will not do. This we will not do.
The energy stored in the pyruvate molecule is extracted in the 5 stages of the Krebs cycle. Click a tab to learn more. Succinate is dehydrogenated by succinate dehydrogenase to form fumarate. In this step, FAD is reduced to FADH2. Next, fumarate is hydrated to malate by the enzyme fumarase. Fumarase is then dehydrogenated by malate dehydrogenase to oxaloacetate. A molecule of NAD plus is reduced to NADH, H plus. This is the last step of the Krebs cycle. The first stage of cellular respiration is glycolysis. In this process, glucose is broken down to form a 3-carbon compound, pyruvate. In the presence of oxygen, the energy stored in the pyruvate molecule is extracted in the 5 stages of the Krebs cycle. Click a tab to learn more.
You all want to note the previous one, or should I take up the next?
I want to know.
To note down, huh? Okay, wait. Because that one was showing the fumaric acid. ये वाला दिखा रहा था. आपको ना सही वाला फिर से देखना पड़ेगा.
Enzyme A enters the Krebs cycle by combining with a 4-carbon compound.
Now this is the next step. Okay.
To form the 6-carbon compound citrate. Citrate is the first product of the Krebs cycle. Citrate is isomerized to D-isocitrate by the enzyme. The first stage of the energy stored in the pyruvate molecule is extracted in the 5 stages of the Krebs cycle. Click a tab to learn more.
Okay, so after oxidative decarboxylation, children, now this one you will be noting.
Enters the Krebs cycle by combining with a 4-carbon compound, oxaloacetate, to form the 6-carbon compound, citrate. Citrate is the first product.
It's not stopping on you.
Citrate is isomerized to D-isocitrate by the enzyme aconitase. Cis-aconitate is formed as an intermediate compound. This reversible isomerization requires a dehydration and a hydration step. Next, isocitrate dehydrogenase decarboxylates the isocitrate to form alpha-ketoglutarate. A molecule of NAD+ is reduced.
It's not getting paused only, children. Wait, let me see. This is the first reaction.
Oxaloacetate by combining with a 4-carbon compound, oxaloacetate, to form the 6-carbon compound, citrate.
So those who want to write, they can write down. Okay, you can, the pause will be done like this only, children. So I'm holding, you write down. This is the first step. The acetyl coenzyme A combining with the oxaloacetic acid or oxaloacetate. In the presence of that enzyme, citrate synthetase, and it is forming the citric acid or the citrate. So CH2CO, COH, then CO, CH2COO. Noting, children?
Yes, ma'am.
तो what you do, इसको ना number 1 डाल के फिर लिखना. Then so you understand कि oxaloacetate फिर citrate में convert हो रहा है. So you put this number 1. Okay, first step. Then I'll take up the next.
Ma'am, Ma'am, what is this H before CO and that highlighted, after that highlighted yellow box?
They are telling that is a coenzyme A only.
Okay,
Coenzyme A. It is the coenzyme A. So you are getting this compound, which is a 6-carbon compound. 4-carbon compound oxaloacetic acid combining with 2-carbon compound acetyl coenzyme A. Should I take up the next one?
yes, ma'am.
Okay. So say quickly, otherwise I am holding it only.
Citrate is isomerized to D-isocitrate by the enzyme aconitase. Cis-aconitate is formed as an intermediate compound.
Now you write out. This is your second equation. So second reaction is citrate getting converted into cis-aconitate. And then cis-aconitate further getting converted into isocitrate. So enzyme needed is same, aconitase. So Neha, you tell me as you complete, say that change.
Okay, ma'am.
And do it faster little, huh? So this way, children, whatever you should understand about the Krebs cycle, the total Krebs cycle in detail you have studied now. Is it clear? Nothing is left in this Krebs cycle. So any exam you want to prepare, you will have to prepare this much. Okay, कुछ नहीं छूट रहा है. अपन ने respiration पूरा कर लिया. Cellular respiration में कुछ नहीं छूटता है. Once you copy these also, so you have understood what is the structural formulas of each of the compound. Okay, not only the name, you know the structural formulas also.
I'm done.
Done. Okay. Now the next.
Requires a dehydration and a hydration step. Next, isocitrate dehydrogenase decarboxylates the isocitrate to form alpha-ketoglutarate. Is reduced.
Now you note down. So here isocitrate getting converted into alpha-ketoglutarate acid or glutarate. It is the same thing. Okay, so it is a dehydrogenation reaction. One hydrogen is removed, accepted by NADPH, NADP, forming the NADPH plus H ions. And carbon dioxide is also removed. Noted?
Yes, ma'am.
Okay. So now we'll take up the next step after this. So you have completed here कि यहाँ से इतना ये cursor से आपको, can you see the cursor? So alpha ketoglutric acid is formed, alpha ketoglutarate is formed. So I'll take up once again now the next one.
First stage of cellular respiration.
Now from here it comes to
is extracted
Here.
in the 5 stages of the Krebs cycle. Click a tab to learn more. In this step, the coenzyme A group from succinyl coenzyme A is removed. The energy released by this reaction is used to form a molecule of guanosine triphosphate or GMT-8. This is known as substrate level phosphorylation.
Okay, so this you note down. So this one step is missing, children. So leave the space. The next one and then you take up this. That succinyl coenzyme A getting converted into succinate. So seeing the cycle, I hope you can understand that which step we are doing. But in the exam, children, you will not write all this. Okay? Simply you will explain the names only. No need to write down these all equations. This is for your other exams. As you
I'm done.
done. Okay. I'll take up once again.
The first stage of cellular, the energy stored in the pyruvate molecule
So this
is
one will tell
extracted
you.
in the 5 stages of the Krebs cycle. Click a tab to learn more. Alpha-ketoglutarate undergoes oxidative decarboxylation to form succinyl coenzyme A. A molecule of NAD+ is reduced.
So this is the previous reaction, children. When alpha-ketogluteric acid getting converted into succinyl coenzyme A. So this way, the numbering you do it correctly, okay? Romisha got it now?
Yes, ma'am.
Okay. Devanshi, are you following the things?
Yes, ma'am.
Devansh,
Yes, ma'am.
are you understanding what we are doing?
Yes, ma'am.
Okay, you missed the classes ना. ये है अपना पूरा Krebs cycle. इस Krebs cycle में how one is getting converted into other, this is explained by this equation. Okay, and which enzymes are needed for these reactions. So you are seeing that specific enzymes will carry out the specific reactions. So where dehydrogenation is going on, where decarboxylation is going on, dehydrogenation होता है तो कोई hydrogen acceptor होना चाहिए और वो acceptor NAD है and that gets converted into NADH. So you can see how it is denoted. The arrows will be made like this. Okay, and here again coenzyme A is used up. So it is forming succinyl coenzyme A and CO2 is removed.
I'm done.
Done? Okay. So now again I'll take up
The first stage of cellular, the energy stored in the pyruvate molecule is extracted in the 5 stages of the Krebs cycle. Click a tab to learn more.
Now this is the last one.
Dehydrogenated by succinate dehydrogenase to form fumarate.
Now note down. Succinate getting converted into fumarate. An enzyme needed is succinate dehydrogenase. So hydrogen removed will be accepted by FAD, which will become FADH2. So it's showing very nicely, children, कि कहाँ से hydrogen निकल रहा है. Okay, so they are highlighting the hydrogens. So one hydrogen coming from CH2, the second carbon, and CH2, that is the third carbon. तो second और third carbon से एक hydrogen remove हो रहा है.
To FADH2. Next, fumarate is nitrated to malate by the enzyme.
Ma'am, done.
Done? Okay.
Fumarase is then dehydrogenated by malate dehydrogenase to oxaloacetate. A molecule of NADH is reduced to NADH H+. This is the last step of the Krebs cycle.
Ma'am?
Yes, Sairam, हाँ बोलिए. Say children.
Ma'am, will you please again show the
previous slide? Okay, I'll just take it once again. Fumarate one you have noted? This one only you are saying, children?
To malate.
Yes, ma'am.
Okay, just 1 minute.
Fumarate. Next, fumarate is nitrated to malate.
Okay, so write down now. This reaction only?
Yes, ma'am.
Okay, so fumarate in the presence of the enzyme fumarase is converted into malate. So here the hydration is going on, that is addition of the water molecule. Noted?
Yes, ma'am.
Okay. So completed all now, children?
Is then dehydrogenated by malate dehydrogenase to oxaloacetate.
This is done.
Yes, ma'am.
The malate getting converted into oxaloacetate or oxaloacetic acid. Okay, so this is the last. That is oxaloacetic acid is regenerated to continue the cycle. So one at a time, remember children, ester coenzyme A will enter and the reactions will be carried out and oxaloacetic acid will be regenerated. And then the second acetyl coenzyme A will come because one glucose is forming two acetyl coenzyme A. So for one glucose molecule, 2 times the Krebs cycle will occur to give the NAD and FAD, which will enter into the ETC.
Of NAD plus is reduced to NADH, H plus. This is the last step of the Krebs cycle.
Okay, so done. Now I'll take up the one video also you can see, children, for this Krebs cycle. So all have noted the things what you had to note? Yes or no?
Yes, ma'am.
हाँ, अभी सारा कुछ clear हो गया होगा कि how what is getting converted into what. So you have got the detailed thing now. Okay, see the video now once.
Krebs cycle. During the process of cellular respiration, an end result of glycolysis will be the production of pyruvate. In the presence of oxygen, pyruvate will enter the Krebs cycle, which is the second stage of cellular respiration. Named after Hans Krebs, who discovered it in 1937. The Krebs cycle is also known as the citric acid cycle, because citric acid is the first compound it produces. Citric acid is a carboxylic acid containing 3 COOH groups. For this reason, the cycle is also referred to as the tricarboxylic acid cycle, or Tca cycle. The Krebs cycle occurs in the mitochondrion and begins with the entry of acetyl coenzyme A, commonly known as acetyl-CoA, which is actually pyruvate that has been converted in the mitochondria. The acetic acid subunit of acetyl-CoA will combine with oxaloacetate, a 4-carbon compound, and form a 6-carbon compound, citric acid, also known as citrate. The enzyme aconitase will convert the citrate into isocitrate. The isocitrate dehydrogenase enzyme will convert isocitrate to alpha-ketoglutarate, NADH, and CO2 will be released as byproducts of the reaction. The alpha-ketoglutarate dehydrogenase enzyme will convert alpha-ketoglutarate to succinyl-CoA. During this step, NADH and CO2 will also be released. The succinyl-CoA synthetase enzyme will convert succinyl-CoA into succinate and will produce one molecule of ATP. The enzyme succinate dehydrogenase will convert succinate to fumarate. During this reaction, flavin adenine dinucleotide (FAD).
Children, are you all following now how the steps are going on and how you have to explain and understand? Devanshi, Rumisha, speak up.
Yes, ma'am.
So it is very clear, I hope now, what is going on in the Krebs cycle. Everyone, Ritu, is it clear to you?
Yes, ma'am.
So how to say that what reaction going on? So in the presence of this enzyme or this enzyme converts this to this. Okay, and then what is released, what is consumed, all that you have to understand. And what is FAD? If you want to note down the full form, you can note down, sorry, flavin adenine dinucleotide. Okay, that is FAD. Should I play again? So these structures are telling you, okay, this is the structure of the different compounds. So very clearly you can see the carbon, hydrogen, oxygen.
Will combine with 2 hydrogen molecules and produce to FADH2. The fumarase enzyme will convert fumarate to malate. In the final step of the Krebs cycle, the enzyme malate dehydrogenase will convert malate to oxaloacetate. During this reaction, NAD will form NADH. The oxaloacetate will condense with acetyl-CoA, once again forming citric acid, and thus the cycle is repeated. Overall, 2 acetyl-CoA molecules are produced from each glucose molecule. So, Krebs cycle will process each one twice, yielding 4 CO2, 6 NADH, 2 FADH2, 2 ATP.
So children, this you can note down. Okay, what is the product till now? You can note down. Glucose molecule giving 2 acetyl coenzyme A. Okay, then 2 acetyl coenzyme A. फिर Krebs cycle लिख दो और Krebs cycle में आपको 4 CO2 मिले, 6 NADH2 मिले, 2 FADH2 you got and 2 ATP. Okay, so this direct ATP is that GTP only. Okay, in your cycle you have written GTP, so it is the same, the ATP. Okay, 1 GTP is equals to 1 ATP. Noted everyone?
Yes, ma'am.
Okay, तो ये summary हो गई कि cellular respiration में क्या होगा.
Summary. The Krebs cycle occurs in the mitochondria. During the Krebs cycle, acetyl-CoA will be oxidized into 4 molecules of carbon dioxide. 6 molecules of NAD will be reduced to NADH. 2 molecules of FAD will be reduced to FADH2. 2 molecules of ATP will be produced.
So is it very clear, children, the Krebs cycle? Yes or no?
Yes, ma'am.
ठीक है, अब कुछ नहीं छूटना चाहिए, means the things should be crystal clear now. No doubt at all. Okay, अब ETC और दिखाना था, but it is already 12 children, isn't it? So it will take time. एक बार this तो you'll have seen, just see this. Okay, wait. (12:30), but the next class will be there, children. It will take time.
Electron transport chain. During the process of cellular respiration, most of the energy is released through a key process known as the electron transport chain. In eukaryotes, this process occurs in the inner membrane of the mitochondria. In prokaryotes, it occurs in the cell membrane. And in photosynthetic organisms, it occurs in the thylakoid membrane. The Krebs cycle, which occurs in the matrix of the mitochondria, produces high-energy electrons, which will be directed to the inner membrane of the mitochondria by electron carriers such as NADH and FADH2. The mechanism of the electron transport chain utilizes these high-energy electrons in the conversion of ADP into ATP. The inner membrane will contain the electron carriers NADHQ reductase, cytochrome C reductase, cytochrome C oxidase, and ATP synthase. In addition, the membrane will also contain the mobile carrier proteins ubiquinone and cytochrome C. NADH will donate 2 electrons and 1 proton in the form of hydrogen atom to the first electron carrier NADHQ reductase. During the transfer, an electron will release energy, causing it to fall to a lower energy level. The NADHQ reductase will absorb the released energy and use it to pump the proton into the inner membrane space. The electron from NADHQ reductase will now bind to ubiquinone, a mobile carrier protein, which will carry the 2 electrons to the next protein complex, cytochrome C reductase. In the cytochrome C reductase, the electron will release energy, which will be used in transferring a proton across the membrane. The electrons from the cytochrome C reductase will bind to cytochrome C, another mobile carrier protein, which will move the electrons to the next protein complex, cytochrome C oxidase. Cytochrome C oxidase will accept 4 electrons. The 4 electrons will react with the final electron acceptor, oxygen, and 8 hydrogen ions. This reaction will yield 2 water molecules into the matrix and release 4 hydrogen ions into the inner membrane. With the movement of hydrogen ions, a concentration gradient will occur across the inner membrane. Another complex, ATP synthase, will release 3 protons from the inner space into the matrix. As the 3 protons move from an area of higher concentration to one of lower concentration, they will add inorganic phosphate to ADP, forming ATP. The electron transport chain will yield 22 ATP molecules, adding to the 4 ATP from glycolysis and the 10 from Krebs cycle. Overall, during aerobic respiration, the complete oxidation of 1 molecule of glucose will produce 36 ATP. Summary. In eukaryotes, the electron transport chain occurs in the inner membrane of the mitochondria. The electrons and protons will be moved across the membrane by protein complexes and mobile carrier proteins. ATP synthase uses protons to produce ATP from ADP and inorganic phosphate. An electron transport chain will yield 22 ATP.
So children, you have seen here what happens in the ETC, how it occurs. So this was the revision of all what we have done in the cellular respiration chapter. अब सिर्फ एक topic बचा हुआ है that Rumisha was asking, so I will be sending that. I said that I'll send that complex 1, 2, 3 and all. Okay, so that I will be sending. अब अभी आपके syllabus के लिए कितना पढ़ना है means for 11th class that you all know. Okay, and whatever further you have studied, that will be for your other competitive exams. So you have everything related to your cellular respiration. So we'll end up the class quickly, children. Okay, I'll stop the recording.